Welcome back. I'm going to read to you chapter 7 on evolution from the book on superenergy. The scientific story for the origin of life is based on Charles Darwin's theory of evolution. His theory is an attempt by scientists to account for the origin of life, in line with the philosophy of the Greek philosopher Democritus, that everything in the universe, including intelligence and life, emerged from the random interaction of material atoms. However, there are problems in this attempt to explain the origin of life, in particular the cell, in terms of chance interactions of inorganic matter. The chance hypothesis for the evolution of life on Earth caught hold in 1953, when Stanley Miller passed electric discharges through a mixture of gases replicating the early atmosphere of the Earth. Amino acids were discovered in the consequent mix, which led the scientific community to conclude that Miller had discovered how life began. They seized on the idea that he had proved life emerged from lightning discharges, which caused the formation of organic molecules from inorganic matter. In line with the predominant materialistic belief amongst scientists, the story that atoms charged by lightning jostle together to form amino acids, which then form proteins, morphed into scientific fact. Scientists argued that over billions of years, these became organized by incremental steps into cells. This all happened by pure blind chance. And the rest is evolutionary history, so to speak. The discovery of amino acids in meteorites supported this story. If life was seeded by the arrival of organic molecules from space, the possibility that life originated by chance was increased by the vastness of space. However, the Miller meteorite mythology is weak as scientists discovered that another molecule called RNA, ribose nucleic acid, is essential in the formation of proteins. RNA is made of small molecules called nucleotides, which are slightly more complex than amino acids. In 2009, nucleotides were fabricated from electric discharges in a lab at Manchester University. But this discovery did not explain how RNA first came to be formed. There is no evidence that RNA can form at random from nucleotides, just as there is no evidence that the appearance of amino acids in Miller's experiment could, by pure blind chance, lead to a fully formed protein. Now, I want you to imagine we were wiped out in some terrible catastrophe and aliens landed on the Earth outside the ruin of a cottage in the Peak District. Unable to explain how it came to be, they began to search for clues. One of them found stones on the ground identical to those in the cottage walls and suggested the stones must have arranged themselves into the cottage by pure blind chance. The aliens said that over an enormous period of time, the random organization of stones could have happened incrementally by the action of wind and wild animals. Another alien pointed out that the mortar holding all of the stone together in the cottage walls was of the same substance as the stones. He wondered how it came to be set so precisely between each stone and how they came to be perfectly positioned in the wall with opening holes just by chance. He reckoned it was beyond the bounds of probability that both mortar and stones could have become arranged together into the cottage walls by luck alone. The stones in this story represent amino acids, and the mortar depicts the nucleotides. The random appearance of amino acids and nucleotides through electric discharges in gases is the equivalent of the aliens finding limestone near the cottage. Just as the random appearance of limestone near the cottage does not explain the cottage, So the random formation of amino acids and nucleotide molecules do not explain how these came to be arranged in the protein and RNA molecules observed in cells. The formation of organic molecules in electric discharge experiments did not prove they self-constructed by chance into complex molecules, or that they did it in incremental lucky steps over immense periods of time. Protein molecules don't just self-construct. They consist of anywhere between 50 and several thousand amino acids that are organized by living cells in a very specific sequence that enables them to perform specialist tasks in cells. On average, 200 amino acids are incorporated in a protein molecule. In every cell, there are many thousands of different types of protein molecule. The chance of even one protein incorporating just 100 amino acids randomly forming on Earth has been calculated at about 1 in 100,000 trillion. Multiply that by every proton that exists. The sequence of amino acids in a protein molecule is set by the RNA molecule. RNA is required to form protein, but proteins are essential to the formation of RNA. So the proteins and RNA would have had to appear simultaneously, perfectly formed, for the first cell to come into being. The chance of that happening by pure blind chance is beyond the bounds of probability. The alien who came up with the chance hypothesis went on to write a book called The Blind Cottage Maker. Though the book attracted a large following amongst the people on his home planet, that didn't prove that what he said was true. It just proved that most people don't think for themselves, but go along with what cleverer people think rather than confront the dilemmas in their own heads and challenge what they read and are taught. The chicken and egg dilemma of life is that new life always comes from existing life. Every cell originates from a cell that has existed before it. Cells divide to reproduce. But the big question is, how did the first cell come into existence? All living cells form into two basic types, a more primitive type called the prokaryotic, and a more advanced type called the eukaryotic. Eukaryotic cells have a nucleus. Prokaryotic cells do not. Most bacteria are prokaryotic. Most of the cells in our bodies are eukaryotic. Most biologists believe that the eukaryotic cell is formed of a number of different types of prokaryotic cells, which came together to form its organelles. That is a sound theory for the origin of the eukaryotic cell, which fits well with the theory of evolution. The question remains, however, where did the first prokaryotic cell come from? The most primitive prokaryotic cell is an extraordinarily complex thing. It is surrounded with permeable membranes made of protein molecules, which are the membrane is many thousand times thinner than the sheet of paper, but far more intricate. Think of the cell as a medieval walled city. Just as the walled city has guarded gates, so the cell membrane also has gates, formed of protein molecules that select what can enter and what can leave the cell. Some protein gates are tubes that allow the passage of only very small molecules, such as air and water. Others are open at one end and closed at the other, with the openings formed to fit a larger molecule of a specific type. When a molecule with the right shape docks into the opening, the gate closes around it to allow its passage across the membrane, while the other end of the gate opens to allow it to enter or exit the cell. The inside of the cell is full of watery liquid containing nutrients and waste, as well as useful end products of the chemistry going on in the cell. Just as the walled city is full of workshops, the prokaryotic cell is full of sites. where very specific chemical reactions occur. Nothing is haphazard. Every reaction is ordered and scheduled according to the needs of even the most primitive cell. Most of the production is concerned with the formation of proteins from 20 or so amino acids. These are used to build the structures of the cell, such as membranes and the enzymes which facilitate the complex chemical reactions. The enzymes are arranged with proteins and RNA to form a ribosome. The ribosome is the chemical factory where the chemistry of the cell occurs. These are the equivalent of the different workshops in the walled city, each designated to a different purpose, i.e., the butcher, the baker, or the candlestick maker. Ribosomes make enzymes, but they can only function with enzymes. How did the first ribosome form? The right amino acids required to form the right enzyme protein molecules must have come together alongside an RNA molecule with the correct sequence of nucleotides. To imagine this occurred by pure blind chance is ludicrous because the math of probability does not allow for that degree of chance encounter. How could so many of the correct molecules come together and entirely by luck join together in the required perfect sequenced combination? It is a story that is hard to believe. We're left with another chicken and egg dilemma. Which came first, the ribosome or the enzyme? In both types of cell, instructions in messenger RNA are received from another complex molecule called DNA, desoxyribonucleic acid, which occurs in a central nucleus in the eukaryotic cell. The instructions in the DNA molecule determine the sequence of nucleotides in the RNA and the sequence of amino acids in the proteins. DNA is a double helix which coils to form the chromosomes in the nucleus of a cell. Even though prokaryotic cells don't have nuclei, they do have DNA double helix strands delivering instructions from their binary code to RNA strands. So there I have a picture of the DNA double helix with the code at the bottom there, and you see it coiling into the double helix, which then coils onto itself to form the strand of DNA known as the chromosome. Right at the top near my index finger there, you see the double chromosome combination that you get in cells. Yet another chicken and egg dilemma is raised by DNA. The formation and replication of DNA requires RNA and protein enzymes, but the formation of RNA and the enzymes requires DNA. The first cell to form would require DNA with the right sequence of nucleic acids to instruct the RNA before the emergence of the protein enzymes. If this happened by chance, there is still the problem of sequence to overcome. The enzymes needed to form the DNA require the DNA to instruct the sequence of amino acids required in their formation. So again, chicken and egg dilemma. What came first, the DNA or the enzyme? Finally, there is the issue of energy. In the medieval walled city, fire was essential for the baker to bake his bread, for the candlestick maker to melt his wax, and for housewives to cook the butcher's meat. But the fire had to be controlled in the ovens to give precisely the right amount of heat. In the cell, energy is required to enable all the many and complex biochemical reactions to occur. Energy in the cell is provided in precise amounts by ATP, adenose triphosphate, operating in organelles called mitochondria. In the cell, these organelles are the equivalent of the ovens in the medieval walled city. Enzymes are needed to produce ATP, but ATP is required to produce the enzymes. So which came first, the ATP or the enzymes? Chicken and egg dilemmas abound in the cell. DNA cannot exist without enzymes, but enzymes cannot exist without DNA. RNA needs proteins, but to form proteins needs RNA. Proteins are made in cells, but cells are made of proteins. The simplest cell is made up of millions of proteins of thousands of different types. All these have to be working together within a selectively permeable membrane for DNA, RNA, and ATP to operate. Walled cities didn't appear on the landscape by chance. They were part of an evolutionary process involving intelligent people progressing from caves and mud huts to towns and cities. People with creative intelligence were behind the evolution of cities over many centuries. Whereas some developments came about by chance, none of them occurred by chance alone. The mud huts and cities were built by people. The existence of towns and cities is proof of the existence of intelligent people capable of designing and building them. Life screams intelligence, but intelligence is not countenanced by the evolutionary theory for the origin of life, because scientists have blind faith in the philosophy of materialism that underpins science. To date, scientists have catalogued some 200 million large fossils and billions of small fossils. Most researchers agree that this vast and detailed record shows that the major groups of all animals appeared suddenly and remained virtually unchanged. There is little evidence in the fossil record to support the theory of incremental evolution. In The Triumph of Evolution and the Failure of Creationism, Niles Eldredge admitted that over time, little or no evolutionary change in species appears to have occurred. He said, "Though I am a fervent believer in evolution and natural selection, I have to admit the evidence for natural selection is very weak." In Sudden Origins by Jeffrey Schwartz, he wrote, "Natural selection may be helping species adapt to changing demands of existence, but it is not creating anything new. The theory that mutations drive evolution is not supported by the science. It's just a story." In Mutation, Breeding, Evolution, and the Law of Recurrent Variation, Wolf Eckhard Lernig of the Max Planck Institute for Plant Breeding Research wrote, "Mutations cannot transform an original species of plant or animal into an entirely new one. This conclusion agrees with all the experiences and results of mutation research of the 20th century, taken together, as well as with the laws of probability. Properly defined species have real boundaries that cannot be abolished or transgressed by accidental mutations." So why do scientists promote the theory of evolution in schools, colleges, and universities as scientific truth when it lacks supporting scientific evidence? Makes you think, doesn't it? Influential evolutionary biologist Richard Lewontin said this is because, and I quote Lewontin, "Most scientists have a commitment to materialism and refuse to even consider the possibility of an intelligent designer. Nonetheless, there are a few scientists who are determined that evolution is driven by intelligence." End of chapter. So I will be coming back with the next podcast on the next chapter, which is The Intelligent Universe, based on a book by Professor Sir Fred Hoyle, who was one of Britain's greatest cosmologists and was one time president of the Royal Society.